Welcome to our next session. I'm honored to welcome Dr. Deepika Polaneni, an associate professor of medicine in the Division of Pulmonary and Critical Care Medicine at the University of Kansas Health System, and associate adult program director for the University of Kansas CF Care Center. Dr. Polaneni's research interests include the identification of non-CFTR genetic modifiers of CF lung disease, as well as a research focus on in vivo transcriptomic and metabolomic studies to identify differential gene expression and metabolite variability associated with CF lung disease severity, with the goal of advancing personalized CF therapies. We welcome Dr. Polaneni, who will present mRNA therapy as a treatment for CF.
Thank you, Jim. I just want to start by thanking Siri and all of the conference organizers and all of our audience today. It's really such an honor to speak with the lineup of speakers that are included in this wonderful conference. And it's really great to be able to come together even in the midst of more challenging times to talk about our joint and committed passion for advancing CF research. So the topic of my presentation today is mRNA therapy as a treatment for cystic fibrosis. And I will say that unlike some of the excellent presentations that have just been completed, this is still such a nascent field in terms of the clinical trials as they're emerging, that I do feel like this is more of a review today with some coverage of the available trial data that we have to date towards the end of this presentation. But I welcome your questions and look forward to engaging with everyone on this new area. So as we get started, really, when we're talking about mRNA therapy, of course, the first and most important question to address is what is mRNA? I think everyone is much more familiar often with talking and hearing about DNA. We use phrases even like it's in our DNA. RNA is actually not so different in some ways. So DNA is made from 4 base pairs that we often hear about, or oligonucleotides rather, cytosine, adenine, guanine, and thymine, whereas RNA is actually quite similar, composed of cytosine, adenine, guanine, but the thymine is changed to a uracil. Otherwise, both share similar backbones where the DNA, D actually stands for deoxyribose and R stands for ribose. So there's a similar backbone to these structures. And DNA is, of course, double-stranded, whereas RNA is single-stranded. The important aspect of why mRNA therapy is attractive in CF and in other genetic diseases is actually because this is actually one way that we can bypass getting into the nucleus when it comes to therapy. So it's taking advantage of the cell's natural mechanisms for normal protein synthesis. And here you can see that the typical function is DNA, which is transcribed to RNA. The coding portions of that are then actually taken to messenger RNA, which is the topic that we are covering in this talk. That is actually then taken out of the nucleus and into the cytosol, where it is then translated into amino acids that form polypeptides. Those polypeptide chains actually then fold into what should be normal functioning protein. And we'll talk about how we can take advantage of that normal protein synthesis process with mRNA therapy today. So to take that kind of long mouthful and make it shorter, what we think of typically is that DNA is really involved in its own replication, but then transcribed to RNA, which is translated to messenger RNA and results in protein synthesis, which is what we want to exploit with these therapies. mRNA therapies in general are seeking to express a protein that is mutated in the patient, non-existent, or underexpressed. So in that manner, we can actually correct for mutant protein, be compensating for a protein that is not present at all, or making up for or supplementing an inadequate amount. And as we've said, this is really using the cell's own machinery. And as we'll get into further here, it's actually using the cell's ribosomes to create the desired protein. So this is a wonderful small review that I would encourage you all to take a look at. The citation is here and later in my references also. But as we look at this process, we see that this is the 5' cap that is endogenously present on messenger RNAs, and it affords some stability to mRNA. And as we move down through that, you actually see the start codon here that is then translated by the ribosome And this is traversed over the process of the messenger RNA until it reaches a stop codon and ultimately to a polyadenine tail, which also affords a degree of stability. Here you can see a few of the typical endogenous factors. This is the poly A binding protein that signals the end, but this is also eukaryotic translation initiation factors 4G and 4E. So these are initiating the process for reading through. What you see on the right panel is actually nanoparticle delivery of a vehicle that's encapsulating mRNA for the desired proteins as this is entering the cell. And you can see that this is a complex process actually for mRNA therapy. The large and negatively charged RNA is actually encapsulated in a delivery vehicle, and we'll talk a little bit more about how that is optimized to be taken into the cell. That's taken into the cell and into the endosome, but actually has to be yielded from the endosome to be able to be delivered and have the vehicle degrade so that the mRNA is available to the ribosomes that are already existing in the cell. And from there, that directs the protein synthesis that we just discussed so that you have a desired protein that ends up staying intracellular or secreted as necessary, or in the case of CFTR, locating to the transmembrane where we want the CFTR to be functioning. There are a number of manners in which RNA therapies can be used to address genetic diseases, but there are actually a variety of ways in which this can be done, and there are gene situations that are more ideal for RNA therapy than others. So for example, in the case of transthyretin amyloidosis, small inhibiting RNA is used, and this is an ideal type of case because this is one gene that's leading to this rare disease and also affecting one cell type. So that's the ideal type of gene therapy to target a monogenetic disease with just one cell type where the expression is primarily carried out. CF is a monogenetic disease, of course. This is caused by genetic defects in CFTR, or the cystic fibrosis transmembrane conductance regulator. However, CFTR is expressed in and affects multiple cell types. So we'll talk a little bit about how that presents some challenges. This actually puts it somewhat in the middle of the spectrum. So we do have a monogenetic disorder, but this is affecting many cell types. And then there's the complex genetic disorders, or actually complex genetic situations such as cancer. And this can be addressed where a package of RNA therapies are actually being delivered in conjunction. Why is mRNA well suited for CF? Well, in general, less than 20% of encoded proteins can be addressed by the use of small molecule therapies. And that's because many diseases are caused by the absence of a wild type protein, or it actually may be resulting of a protein that lacks an appropriate binding site for the mRNA, for small molecule therapies. In the case of cystic fibrosis, we now know that global estimates are reaching up to about 80,000 people with CF. And although CFTR modulator therapies have tremendously advanced treatments in the recent era, there remain at least up to 10% of people with CF in the United States, according to the currently available registry data, that are ineligible. And that may be because of mutation status that is not amenable to small modulator therapies for both of the reasons that are cited up above, or it may be because of the aspect of having adverse events to such therapies. So these are cases in which mRNA therapy may be more attractive. As we talk about mutations that may or may not be eligible for small molecule therapies, that we actually then recognize that not all CFTR genetic mutations are the same, and therefore they are not all amenable equally to small molecule therapies. So here on the left, you can see the normal process of transcription that is taken to translation, as we discussed, resulting in a normal protein, and that normal CFTR protein is trafficked to the cell membrane, and here you have normal chloride trafficking in the healthy situation. There are several classes of CFTR mutations, and they are each amenable to different forms of therapy depending on the mutation type. So here for class 1 mutations, this is the classic situation in which mRNA therapy may be helpful. Here you see the mutated CFTR protein is transcribed but results in unstable mRNA, which is broken down and results results in no protein at all. You can see how delivery of normal CFTR mRNA would be very desirable here. In class 2 mutations, such as the Delta F508 mutation, or that mutation which is present in at least 70% of people in the US, the mutation results in a misfolded CFTR protein that still results in no chloride trafficking because that misfolded protein is then degraded and is unable to make it to the cell surface. For class 3 through 6 mutations, there is a variety of different aspects of faulty result of the protein that can yield reduced gating, faulty channel, inadequate surface of CFTR presence, or a high turnover of CFTR at the cell surface. The good news in many ways is that for class 2 through 6 mutations, the recently approved CFTR modulators do have therapeutic application and potential in many, but not all cases. So in the case of class 2 mutations for the Delta F508, for example, the combination of elexacaftor and tezacaftor aids in the protein misfolding, and that results in a protein that does make it to the cell membrane. And the addition of ivacaftor then allows the protein to function normally at the cell surface. Similarly, ivacaftor has been approved for about over 38 mutations, most of which are in the class 3 area here. And that's how you can see that these are treatable by potentiators or the combination of correctors and potentiators, as we were just discussing. However, mRNA still remains attractive, as we were discussing here, for class 1 type mutations, and also for a number of mutations that may span across these other classes, because we know that there are still yet rare mutations that may not be addressed by these small molecules. So rare and ultra-rare mutations are absolutely one of the most important reasons that we do think about mRNA therapy. And we know that there are more than 1,000 CFTR variants that are demonstrated in less than 5 patients each worldwide. And furthermore, we know that low numbers, such as N of 1 studies, may not always be feasible to be able to show the advantage or benefit of a small molecule therapy such as CFTR modulators. So mRNA therapy remains a very attractive option to pursue. We hear a lot about nucleic acid therapies today. And so when we talk about mRNA therapy, gene therapy, and gene editing, it really becomes the question of what's in a name and how are these aspects different from each other. So one of the important distinguishing factors is that mRNA therapy uses the cell's existing machinery to produce functional protein and does not enter the nucleus, whereas gene editing is actually correcting the genetic defect at its source by editing the DNA. And gene therapy delivers the desired gene or DNA by a viral vector, such as often discussed adeno-associated virus. So there is absolutely distinction between these 3 different types of gene therapy. There are also differences that are very important in terms of the mRNA action between these types. So there's the aspect of permanence versus semi-permanence towards the treatment. DNA-based therapies may provide long-term expression of a gene, as this is actually affecting the genetic code itself, whereas mRNA therapies are transient and they do require repeated dosing. Now that poses its own advantages and disadvantages, as we'll cover in a little bit. There's also the aspect of delivery. So DNA-based therapies may be delivered by adeno-associated viruses, as we just discussed, but mRNA therapies are delivered more commonly by lipid nanoparticles that may be proprietary. So as we recently just discussed, the RNAs are quite large and negative in charge. So the enveloping of these RNAs in a lipid nanoparticle, as is the case here also with polyethylene glycol, actually helps in that endosomal process to be taken up by the cell and helps to facilitate the transport of the mRNA into the cell, where it needs to then be degraded by the endosome and made available for the cell's own machinery to create the functioning protein. There are a number of advantages with mRNA. There's the therapeutic potential of the intracellular, transmembrane, and secreted proteins. There's also the fact that this restores gene expression without the risk of genomic integration. As we mentioned, it does have drug-like behavior with ability to repeat dose and adjust the dose or dose interval. There's also the factor of endogenous post-transcriptional modifications that increase the probability of correct folding, glycosylation, and trafficking of the protein and decrease the risk of immunogenicity, and we'll cover that in a bit more detail. There are minimal changes to the manufacturing process for multiple targets. And so that's an attractive aspect from the synthetic standpoint. And finally, there is also the potential for rapid development from a target gene selection to the product candidate. However, there are also a number of challenges that arise with mRNA therapy. So mRNA is susceptible to degradation that may be amenable by some of the modifications that can be made that we'll cover. There's also the aspect of delivery. So lipid nanoparticles must be tissue specific and biodegradable. And there's also the concern for immunogenicity. So there is the risk of activating the immune system via toll-like activity and retinoic acid inducible gene-like receptors. There's also concerns for manufacturing, as it can be difficult to achieve high quality and high pure mRNA with the scalable manufacturing process. So there are certainly pros and cons in both directions. One of the challenges that's unique, or a few of the challenges that are unique to consider in mRNA therapy and CF specifically, include the thick mucus barrier. So this is actually a scheme that is blown up from the what we would expect to see in the airway lumen. And we see here that there's the thick mucus barrier. There's also the consideration of which cells. So the superficial respiratory epithelium are where we typically have the diagnosis of CF. And we know that CFTR is certainly expressed there. But we also know that the submucosal glands are a critical component of this portion of the airway lumen that are producing CFTR as well. So the ability to actually get the lipid nanoparticle past this thick mucus layer and into the correct cells is a remaining challenge, and it is yet to be determined in further studies. There's also the question of how much is enough. So there is some demonstration in animal models and in vitro that having even up to 5 to 10% of wild type CFTR may be sufficient to cover the amount that is needed to create a more normal physiology type for people with CF. There's also the challenges about the fact that CF is a multi-organ disease. So while the current studies are focusing towards the respiratory delivery of CFTR mRNA, the question remains, what about the rest of the individual? So we know that the average life expectancy in CF is now greater than 40 years, and that's even before CFTR modulator therapies. And this has largely come about due to treatments that manage bacterial infections, replace pancreatic enzymes, enhance nutrient absorption, and yield the chronic chest clearance of mucus. What happens when we're addressing mRNA therapy that is targeted primarily towards the lung? These are really focused towards the lung because we know that pulmonary disease and CF is the leading cause of morbidity and mortality, but non-pulmonary manifestations of CF will still remain a challenge that we will need to address through traditional methods such as pancreatic enzyme replacement therapy and exocrine pancreatic insufficiency, vitamin replacement and intestinal absorption of fat-soluble vitamins, insulin or oral hypoglycemics for CF-related diabetes, and as best as we have it at this time, ursodiol for cystic fibrosis-related liver disease. There will also be the question of the potential need to have ongoing treatment or management of bacterial infection, as we know that bacterial infection may recur in the setting of CFTR modulators, and as we well know that bacterial infection remains a chronic issue in people who have non-CF bronchiectasis. We've come a long way in mRNA research, so I think it's a nice juncture to look at where have we come from and where are we going. So turning good into better, one of the topics that we've touched on so far is chemical modifications that can improve the efficiency of RNA drugs by at least 3 potential mechanisms. So there's the ability to decrease immune activation. There's also the opportunity to increase drug stability, and there's the opportunity to enhance the duration of effect. Finally, also to influence the affinity by which the RNA binds other nucleic acids and proteins. So for chemical modifications that are made to mRNA drugs, these are often made at the positions in the mRNA that are already naturally modified in our bodies in the endogenous eukaryotic mRNAs. So for example, endogenous mRNAs are modified in human cells to include a 5' cap, and mRNA drugs are also modified at the 5' end. These 5' cap modifications have been used to decrease activation of innate immunity and can often be divided into 3 different types. So there's cap 0, cap 1, and cap 2. And what this means is the addition of a hydrogen or a number of methyl groups to the 5' end. And that can actually increase the stability of the mRNA and decrease the innate immune activation. There's also the potential for modification to the poly A tail that we covered at the very beginning of this lecture. So scientists have actually used a technique called tail-seq and identified that different lengths of the poly A tail can confer different amounts of stability, and looked at the optimal length that affords the best half-life, and have found that 50 to 100 is actually conferring a more stable half-life. And finally, there are changes that can be made to coding sequence modifications. So the addition of a pseudouracil, for example, or any of the changes that are noted in this third row can actually decrease some of the potential risk for immunogenicity. How have these types of modifications been translating into practice? Well, most of the discussion of the actual modification for mRNA therapies is, of course, proprietary to different companies. We can look so far at the experience that we have from the clinical trials that we know about to date. So I'll cover that briefly here. And although there is a paucity of what is publicly available, we've mined what we can for this discussion. So the first of these was actually QR10, which completed a phase 1b open label multi-center study that was exploratory. And this was done to estimate the effect of intranasal delivery of QR10 on nasal mucosa in restoration of CFTR function. The primary endpoint in this study was the within subject change from baseline in total chloride transport. And this was measured by the nasal potential difference, or that's actually a direct measure of the change in the short circuit current or ion channel transport in the respiratory epithelium, in this case, the nasal epithelium. And this was done in adults who were either homozygous or had at least one copy of the Delta F508 CFTR mutation. This was completed in 2016, and what is available is that the company subsequently went into a partnership with Galapagos to combine some of those technologies. The trials that we know most about to date actually come from Translate Bio. So this slide is adapted and was afforded courtesy of Translate Bio. Here, this is again, you see the classic structure of the 5' cap, the untranslated region, coding region, 3' untranslated region, and poly A tail. And this is representative of the proprietary lipid nanoparticle delivery from Translate Bio. So this is the formulation of MRT-5005, and that's a codon optimized human CFTR mRNA, of course. And this is delivered via nebulization to the lower respiratory tract directly into the lungs. So Restore CF is a phase 1 and 2 clinical trial that has been conducted in adult patients with CF in the United States. And the results of the first interim analysis were presented in 2019, and published in the Pediatric Pulmonology Supplement for the North American Cystic Fibrosis Conference. And so I'll cover those here as those are published and available. But after a single ascending dose study that comprised data through 1 month of follow-up after a nebulized dose of MRT-5005 or placebo, the results showed that MRT-5005 was generally well tolerated at low and mid-level doses. The lipid levels in blood in some, but not all of the patients did indicate that there was delivery through the mucus and was making it into the cells, as this was reflected in the bloodstream. And 4 of the 9 patients had an increase in the percent predicted FEV1 within 8 days after treatment that was higher than expected based on known variability of percent predicted FEV1. The top line single ascending dose and multiple ascending dose data are expected to be presented in future, but have been shared in a press release, which I would encourage you to pull. And that was released in March 2021. As that has not been formally published, but is publicly available, I'm just summarizing that here. It similarly showed that there was general good tolerability, although there was one serious adverse event of pulmonary exacerbation in the single ascending dose and one hypersensitivity reaction. However, there were no concerns for immunogenicity in either the single ascending dose or multiple ascending dose arms. There was no significant change that was observed in the percent predicted of FEV1. However, Translate Bio has been moving into a next line of studies using its next gen mRNA therapies, and that is something that the community is eagerly anticipating. Arcturus has been in preclinical development of ARCT-032. This is using the proprietary LUNAR-CF platform in cystic fibrosis. So this is a lipid mediated delivery. And this is also using the self-transcribing and replicating mRNA or STAR technology. And this is using the innovative manufacturing of the mRNA drug substance and the LUNAR formulated drug product. There is little available publicly at this time, but this remains in preclinical development. And finally, we know that Vertex and Moderna have partnered. This was first announced in 2016, that an exclusive collaboration was developed to work on the mRNA therapy in CF from the combination of these two companies. And more recently, there has been a second collaboration that's been announced between Moderna and Vertex to look at proprietary mRNA and lipid nanoparticle technology for the potential for gene editing. So this is actually an application where mRNA can then be used to actually translate to endonucleases that can aid in the gene editing process. We've heard a lot about mRNA in the news in the past year, especially during the pandemic. As Dr. Moss so nicely covered, we have learned quite a bit about the potential for mRNA vaccines. So this is actually two of the co-founders for just one of the BioNTech formulation of mRNA vaccine for SARS-CoV-2. We have seen through that there's been increased attention drawn to mRNA, including the potential for vaccine development in cancer. And this is a French news outlet. And finally here, one of my personal favorites, a poster that indicates that the mRNA vaccine is like a most wanted poster that tells your body how to recognize COVID-19. It can't and won't change your DNA from Waterloo. So these are just some examples of how widely mRNA is now being discussed around the globe. The advent of mRNA vaccines has proven, as covered earlier today, incredibly efficacious beyond what we could have expected in the novel coronavirus SARS-CoV-2 that has been causing the global pandemic from 2019 November to present. This resulted in vaccine development that has been given emergency use authorization by the FDA, and the vaccine first became distributed in December of 2020. This is representative of those two very successful coronavirus vaccines that are using these lipid nanoparticles to deliver messenger RNA into the cells. And once the mRNA is directing the cells to actually produce the SARS-CoV-2 spike protein, so that the body's immune response is then elicited. And when the body is presented with the actual SARS-CoV-2 virus, it's already recognized and the host is able to develop an adequate immune response. Some of the safety concerns that remain barriers are, in fact, immunogenicity. Unfortunately, with the SARS-CoV-2 vaccines to date, we have seen more reactogenicity, which we believe to be reflective of a response to the lipid nanoparticles, because those same responses can be seen in animal models, even when mRNA is not included in these lipid nanoparticles. So this can amount to things like myalgias or muscle aches, severe fever, headaches, and oftentimes this is being seen after the second dose, which is actually a good sign that the body is mounting an immune response as we would expect. For immunogenicity, however, this would, in the case of CF mRNA therapy, be the concern for looking at anti-CFTR antibodies or T-cell sensitization to CFTR, which could cause an adverse immune response in the host and potentially be limiting to the ability to continue treating an individual with CFTR mRNA therapy. So this second slide is also courtesy of Translate Bio, and I think a nice summary that really shows how the lipid nanoparticle that's encapsulating mRNA of CFTR enters the epithelial cell. This is getting across the cell membrane once it's already made its way through the mucus barrier. The lipid nanoparticle then degrades, the mRNA is released, as you can see here, and is so important to emphasize, this does not get into the nucleus and does not change the genetic code. Rather, it's taking advantage of the cell's existing machinery, and the ribosome is translating this mRNA, which is then producing normal CFTR protein, and that CFTR protein is able to function at the apical membrane of the cell as it's intended. And this is all, in this case, through nebulized delivery, and as we've discussed, intranasal delivery has also been explored. I think as we move into the future, the potential for mRNA therapy remains very bright. We know that the current landscape of clinical trials has certainly been paused in many ways due to the coronavirus pandemic, but the number of opportunities to be able to explore mRNA therapy, whether that's for the direct delivery of wild-type CFTR, or if that's actually to aid and augment the potential for genetic editing, remains a promising future ahead of us in mRNA treatments in CF. So I think we're actually a little bit ahead of time, which will leave us plenty of time for questions and to make up for any slow earlier.
All right. Thank you, Dr. Polaneni, for that incredible presentation. And you're right, it is time for questions. Siri?
Hello. Thank you, Dr. Polaneni. That was such a phenomenal presentation. So clear. And of course, we have questions for you. So starting from the top, from Linda Bowman, can mRNA therapy take place at the same time as phage therapy?
That is a great question. And actually, I don't know if we have the ability to have our additional panelists jump in from our phage background. I'll have to actually concur with John and see if we can get a response back jointly, because that's actually not something that I have looked at.
Would post-transplant people be eligible for mRNA therapy?
So at this time, there is no indication that we would have that suggests that post-transplant individuals would be ineligible to participate in receiving mRNA therapy. However, one of the barriers for that is that because the immunomodulatory treatments that are used in transplant will invariably modify that milieu, it is not expected that they would be part of clinical trials, just as they typically have not been part of clinical trials in the past. So the actual implications to use this as a treatment for individuals who have been transplanted will be, we'll know more about that as we know potentially more about drug-drug interactions and as we begin to start seeing it used in practice.
Let's see, Gabriella asks, would the immunogenicity be a problem for people with CF who already have infections and or are post-transplant?
So there is really no specific concern for immunogenicity being a product of increased infection in CF. This is actually a process that's quite specific to the delivery of the mRNA therapy and or the vehicle that it's being delivered in. So the increased immune response that is ongoing for people with CF in terms of the constant immune response against bacterial infection does not necessarily impose an increased immunogenic response against CFTR therapy.
Thank you. And Missy wanted to know, how often would patients need to receive this?
So the treatment doses for the MRT 5005 trial that has been published, those show a range of anywhere from weekly to there has also been consideration for daily treatment. The frequency of treating the individual with the mRNA therapy entirely depends on the stability of the individual formulation of the mRNA therapy by the different companies.
And just tagging on with that, so there's the frequency. How long does it take to actually inhale this therapy?
That also has a significant variability depending on the company, on the type of drug that has been formulated. So that has to do with a number of factors, including the time over delivery that the drug can be adequately deposited. It has to do with the size of the therapy. So there is certainly a range and variation. The intranasal delivery is shorter, but then we also know that the intranasal delivery versus nebulized or bronchial deliveries all actually need to be assessed in the independent tissue types.
Can increased IgE inflammation or inflammation due to ABPA hamper the effectiveness of mRNA therapy?
So I don't know the answer to that specifically in terms of antibodies that are present due to ABPA. However, there is some evidence that decreasing inflammation may actually augment the efficacy of these mRNA therapies to be able to be taken up.
From Julie Dash, can you discuss the idea of systemic delivery via injection, similar to what is being done with the COVID vaccination?
So the first, hi to Julie, and second, the delivery of mRNA systemically encounters a number of issues that may arise. So there's the potential for off-target effects. There's also the factor that the systemic delivery is actually being further explored more so in the case of like stem cell therapy and not as much in mRNA therapy for CF disease right now.
And I just lost what percentage of patients actually develop immune responses to mRNA therapy, and how severe is the response?
So because the clinical trial data that we have currently is very limited, we only really have the tabular data that's available. And so far from the MRT 5005 trials, there was not evidence of immunogenicity in the single ascending dose or multiple ascending dose arms.
From Beata Illich, are there ways to achieve cell type specific delivery?
That's a fantastic question. I can't claim that I personally know the answer to that, but that is actually one of the challenges for mRNA therapy in CF, as we were discussing. So there is certainly the superficial respiratory epithelia that we wish to target. There are the submucosal cells. And now we know more through single cell RNA-seq about ionocytes and their production of CFTR, although the exact role is still being further elucidated and understood. So at this time, I am not aware of cell-specific delivery within the respiratory tract, but there certainly is the concern that one needs to consider for off-target effects, for example, on pulmonary immune cells such as macrophages.
And I will say that question was asked by Beata Illich, who in her own right is a brilliant CF researcher. So no wonder, that was a great question. So we have a question from Carissa. Why is the delivery of the mRNA within lipids considered to be proprietary?
Well, I'm trying to think of the best way to explain that. You know, the way that these nanoparticles are taken up is going to confer some level of the efficacy ultimately. So having an optimized vehicle that the cell can take up and be able to use wild type CFTR will be dependent on each company and will be tweaked and modified to try to get the best optimization of uptake. So that's why that's proprietary across the different companies.
And before I ask the next question, there's been a request that you stop sharing your screen.
Oh, okay.
Just so that people can see you in your glory.
That's great, because then I might actually see the chat and not stammering quite as much.
Let's see. So we have a question from Adam Gulper. Since Translate Bio says it's now preparing a next-gen mRNA therapy, is it suspected that MRT 5005 will end at phase 1-2 and a new program will need to start?
I certainly would not be in the position to speak for Translate Bio. They do have a press release that's available from the March 2021 update. So I would certainly encourage Adam to check there.